Ladies and gentlemen, I am delighted to welcome you to the Metabolomics Society's first webinar. Hosted by the Early Career Members Network, we have been working hard to create a webinar series dedicated to knowledge transfer, training, and the development of our early career scientists within the metabolomics community. We are planning to host at least 6 webinars this year, providing a combination of short and long-duration seminars in a broad range of topics, covering 4 target categories. Number 1, back to basics. Number 2, informal training. Number 3, debating sessions. And finally, number 4, our ad hoc sessions. Part of the 4th category is dedicated to more prominent and expert speakers in metabolomics, providing the audience with presentations on current topics as well as further insight into the speaker's own research. Dr. Oscar Yanis is in fact our first speaker of this category. Now, before I go on to introduce our speaker, I would like to inform you that all material will be recorded and uploaded to the Metabolomics Society website, allowing members of the Society access to the live content at their own convenience. However, given that this session is free, courtesy of the Metabolomics Society, you will be able to have access to this webinar online once it has been completed. So do make sure to keep an eye out for our updates on the Society's social media sites. For today's session, Dr. Oscar Yanis, who is the Principal Investigator of the Center for Omics Sciences and Assistant Professor at the Rovira Virgili University in Spain, will give a 20-minute presentation entitled Metabolomics, only suitable for multidisciplinary teams, that will not only cover the complex nature of metabolomics practice, but provide insight into his group's research and experiences. Please note that there will be time for questions at the end on a first-come, first-served basis. So once the presentation has been completed, send in your questions via your webinar control panel application. Over to you, Dr. Yanis.
Thank you, Eva. It is an honor to inaugurate the seminar sessions of the Metabolomics Society. I would like to take this opportunity to address early career members of the Metabolomics Society and in general PhD students, postdocs, and researchers with little time in the field of metabolomics on the importance of multidisciplinarity. This is, of course, a personal view as a result of 7 years of conducting metabolomics studies. Among the omics sciences, metabolomics is possibly the most multidisciplinary of all, involving knowledge on electronic engineering, statistical physics, analytical and organic chemistry, biostatistics, and of course, biochemistry and metabolism. In later slides, I will present an untargeted metabolomics workflow that provides examples of this multidisciplinarity. Yet, the multidisciplinary nature of metabolomics begins here. Genes, transcripts, and proteins result from the concatenation of a small set of known monomers, namely nucleotides and amino acids. And existing technologies and bioinformatic tools allow for the amplification and accurate characterization of the sequence of monomers. And this is partly thanks to the correspondence between triplets of nucleotides and amino acids. Metabolomics, in contrast, aims to identify and elucidate the structure of metabolites, which are not sequence of monomers and do not result from a residue-by-residue transfer of information. Instead, the large diversity of metabolites in living organisms result directly from series of chemical transformations catalyzed mainly by enzymes. As a result of a wide range of polarities, molecular weights, different functional groups, and chemical stability and reactivity, metabolites must be detected using several technologies and instrumental setups to increase metabolome coverage, which is something remarkable that does not happen in genomics and proteomics, at least to the same extent. As a result of this diversity of analytical platforms, converting raw spectrometric data into biological knowledge has become the major bottleneck of metabolomics. And for groups like ours, this is a very important issue, since we don't do metabolomics to do signal processing and multivariate statistics alone. We do metabolomics to study metabolism. So with this information in mind, I will go through the different steps of an untargeted metabolomics workflow, hoping that you will recognize the multidisciplinarity involved. So the first step in this untargeted metabolomic workflow is experimental design, which is something that sometimes is underestimated. But briefly, my message is that you should avoid this type of experimental designs. Probably some of you will feel represented by this slide, and this is a common situation that I typically face with some of my collaborators. This is not an experimental design, but this happens, and this is common, particularly with clinicians. When collaborations start with clinicians, they have a lot of samples in the -80, and they just want to do metabolomics and find out something. Avoid this type of experimental designs. The second step is sample preparation. Sample preparation, there is a key message about sample preparation and metabolite extraction. There is no single best extraction procedure for metabolome analysis that covers all intracellular metabolites. And this is a reality that we must accept, regardless of the combination of solvents, the polarity of the solvents, the temperature, and the pH. Regardless of the combination of these three parameters, you will not find any single procedure that can extract all intracellular metabolites. The next step, once we have extracted sample preparation, is sample analysis. I will focus on mass spectrometry, and this is the basic diagram of a mass spectrometer. So the sample can be introduced, as you know, in three different options: as a gas phase using gas chromatography, liquid phase using liquid chromatography or capillary electrophoresis, or in solid phase using surface-based approaches. Here is an example of the importance of liquid chromatography, and in particular the mobile phase of the liquid chromatography. Here we compare the ionization efficiency of a large number of metabolites extracted from E. coli using ammonium fluoride or ammonium acetate in the mobile phase of the chromatography using negative ionization electrospray. As you can see, the intensity of all these compounds is increased relative to ammonium acetate. And in fact, there is a large number of compounds that were only detected when ammonium fluoride were used in negative ion modes as compared to ammonium acetate. The second phase of the ionization process of a mass spectrometer is the source. So the compounds can be introduced as gas phase, and these can be ionized by electron ionization, chemical ionization, or if they are soluble, they will be ionized typically by APCI or electrospray. And solid phase ionization, the most characteristic or the only one, is laser desorption ionization. Each of these ionization sources produce characteristic ions that, as many of you will know, need to be processed. The data generated from these ionization sources need to be processed in different ways. I will give you an example of a solid base approach, which is nanostructure initiator mass spectrometry. This is a technology that was developed at the Scripps Research Institute, in which I collaborated, or I helped to develop. And this is a technology, a surface-based technology based on nanostructure material. And the most characteristic application, or the most probably appealing application of NIMS, is for tissue imaging of metabolites. So one can complement the typical metabolomics studies in which tissue is disrupted and homogenized with this technology to localize the distribution of particular metabolites. Now, in my group, we are developing novel nanostructured materials for tissue imaging. In particular, we are using sputtering the position of gold, silver, and tungsten oxide nanoparticles on top of the tissue to do tissue imaging experiments. This work will be presented in June at the Metabolomics Society. But as a demonstration of this multidisciplinarity, sputtering is used extensively in the semiconductor industry to deposit thin films of various materials in integrated circuits. So this knowledge that is very common knowledge in a department of electronic engineering has been used to implement, or has been implemented for this particular application in metabolomics. So next, once we have ionized our metabolites, these metabolites can be separated using different analyzers. Most typical analyzers are quadrupoles, time of flights, ion traps, and orbit traps. And you will agree that each one of these analyzers will also have particular features like different resolution, different mass accuracy, etc. So these will all affect the data processing later. So the next step is data processing, or probably you can also call it signal processing. Here I will show you an example, an NMR example in this case, in which we have developed 2D diffusion order NMR approach to characterize lipoprotein subclasses by applying the convolution techniques, and by applying these deconvolution techniques, we are now able to calculate the size of lipoproteins and the number of particles of VLDL, LDL, and HLDL lipoprotein classes. This is a nice complement to classical lipidomic studies in which lipids are extracted and analyzed individually. But as you know, lipids in the bloodstream travel through lipoprotein particles. So one of our goals now is to complement classical lipidomic studies with the characterization of lipoprotein classes. So next, once we have extracted, analyzed the sample, and processed the data or the signals, the spectral signals, these signals must be identified in order to associate this particular signal to a metabolite name. So the easiest or the most typical scenario is this. So we have a biological sample and we have a parent ion with its tandem MS spectra. And this tandem MS spectra is compared against a standard, a commercial standard. If the tandem MS spectra match with the standard and the retention time is the same, we can unequivocally say that we have identified this metabolite. However, there are increasing evidences that the number of unknown metabolites is very large, which is something that should make us reconsider our knowledge of classical biochemistry and cell metabolism. So in this particular scenario where we have to identify an unknown metabolite, we have three different options. The first one is NMR characterization. However, this option is limited by the amount of purified compound, which is something that probably can be obtained when working with large amounts of materials like plant materials and urine with large volumes that can be obtained. but it's relatively difficult to isolate unknown metabolites from cell cultures or small pieces of tissue, which is a very common material or matrix, biological matrix in metabolomics. So the second approach is to use MSMS data from these unknown metabolites to get structural information. So in our lab, in collaboration with a group of physics, we have developed IMET, which will also be hopefully presented at the Metabolomics meeting. It's a computational tool for a structural annotation of unknown metabolites. So IMET, for each unknown metabolite, the algorithm takes as input its experimental MSMS spectra for different collision energies, its exact mass, and its isotopic pattern. Then IMET compares these inputs to a reference database containing experimental MSMS spectra of known metabolites. The algorithm outputs a sorted list of candidate neighbors of the unknown metabolite. For each candidate neighbor, IMET gives a Bayesian transformation that converts the candidate into the unknown metabolite, and the accuracy of this prediction is defined as a numeric score. Finally, or eventually, at the end, we will always need a chemist to synthesize this novel compound. And I'm showing you a nice example of this particular case. So this was a publication in JACS, and that was a collaboration that I lived because I was at the Scripps Research Institute when this work was done. This was a collaboration between Barry Sharpless lab, which is one of the most famous chemists worldwide. It's a Nobel Prize winner, and Gary Seuss lab, which is a very well recognized investigator in the metabolomics field. So in this study, they were able to find a natural compound that was upregulated in a microorganism that was adapted to live at high temperatures. When they performed MSMS experiments on this natural product, they thought that they were identifying N-acetyl thermospermine because that was the match on the database. However, the fragmentation pattern of the natural product was slightly different from N-acetyl thermospermine, and they realized that they were dealing with a different structure. So after 3 rounds of chemical synthesis, and by round I mean after 3 tries of structure, so they synthesized structure and they found it was different. They then synthesized another structure and they found it was not exactly the one that they were observing in the natural source. On the 3rd synthesis, they were able to synthesize this novel and completely unknown metabolite that was not reported in any book or any database, and reported as a new metabolite synthesized in this microorganism. Finally, after all this workflow has been performed and the metabolites have been identified, the next step is to contextualize all this information in a biological environment or context. So for this, we have different options. Typically, we have a static picture of metabolism. So we have information about which metabolites are up or down regulated in our metabolic map. Can we extract, can we generate hypotheses about the activity of this cell or this metabolic network? I like to use this analogy. This is the city of Barcelona, and if I want to understand the activity of the city, if I want to know how this city works, where people go to work, what type of areas are more busy, etc., Can I say something about how the city of Barcelona works if I know the number of cars that are here in these parking lots? If I tell you that today we can find 5,000 cars here, and in a week from now, at the same time, we only find 200 cars, can you say something about the activity of the city just by knowing the number of cars here? Probably not. Probably you are very limited if, about the rest of the city. So that's what brings me to the next slide. There is another type of information that can reveal more details about the city of Barcelona or any other city, and this is traffic. So if we know which roads contain more or less traffic, the amount of cars that are on the roads at different times, and which areas have more dense or light traffic, we can probably know which areas of the city contain more industry, which are more areas that are for fun in the evening, etc., etc. So with this in mind, in our lab, we have developed two different tools, one based on mass spectrometry and another one based on NMR. George is an algorithm that we have developed for unbiased tracking of isotopic labels in LC-MS profiling. This work will be presented also at the Metabolomics meeting. And very briefly, here the goal is to use LC-MS profiling data from unlabeled samples and labeled samples, and being able to identify which compounds contain labeled carbon from the original precursor. So in this example, cells were incubated with unlabeled glucose and labeled glucose. And here you have an example of ADP glucose in the unlabeled sample and ADP glucose that is unlabeled, and the +5, which corresponds to the label of ribose in the structure, +6, which corresponds to the label of glucose, and +11, which corresponds to a population of ADP glucose that contains both glucose and ribose labeled in the same structure. The second approach that we have developed is proton elucidation for isotope analysis. Probably some of the ones that have kids will notice the names of these two tools, George and Peppa. So we have a period in the lab with quite a lot of kids and conversation about kids. So Peppa is a 1D proton NMR approach that we have developed to follow or to track carbon labeling. The details of this approach will be also presented at the Metabolomics meeting. And here you have an example or an application of Peppa in which we incubated cells with labeled glucose or labeled glutamine, and we were able to track the fate of these labeled glucose, of these labeled carbons on downstream metabolites. In some examples, we were able to find populations like this, the case of aspartate, in which there were a population that contained carbons from glucose and carbons from glutamine. So finally, our ultimate goal is to integrate the static picture of metabolism using MS and NMR technologies with the dynamic picture of metabolism by using isotope labeling, and the integration with other omic sciences, particularly proteomics. In order to do that, we are collaborating with the statistical physics, because physics and the statistical physics in particular are used to work with complex networks, and they have built a great knowledge on graph theory, and they have this ability to understand complex networks, and metabolism is an example of complex networks. Finally, I would like to thank all our collaborators. As you can see, as you can see here, most of them comes from the electronic field, in physics, in statistical physics. We also have collaborations with clinicians. There is one very nice work that we are conducting right now, in which we are integrating quantitative proteomics, static metabolomics, and dynamic or flux analysis in collaboration with Shabazz Mohammed and Albert Heck from the Netherlands Proteomics Center. And of course, I want to thank the people in my lab for the hard work behind all these studies and developments. So you will see most of them at the Metabolomics Society meeting in June, and most of the stuff that I have presented here will be presented at the meeting. Thank you very much for your attention, and now I'm open for questions and discussion if needed. Thank you.
Thank you very much for your lovely presentation. Now, ladies and gentlemen, if you can please send in your questions via the application of your control panel. And we already have a question for you, Dr. Yanis. How important is it to have better instrument capabilities versus better research hypothesis for large-scale metabolomics projects?
Yeah, that's a good question. You definitely need instruments with good accuracy and good resolution. That's a reality. However, even if you have the best instruments, without a clear hypothesis or without a clear or good experimental design, I don't think this study is going to be successful. I honestly think that even having not so good instruments, but a clear study and a clear experimental design, you have more chances to be successful, because in fact instruments have been improved over the last years. And when we started, or I personally started doing metabolomics in 2006, 2007, instruments were not so good as right now, and still we were able to obtain interesting information about cell metabolism. So probably it's more important to have a good study, a good experimental design than state-of-the-art instrumentation.
Okay. Our next question. Can large-scale metabolomics projects be standalone without support from basic biological experiments and other omics approaches?
Can you repeat the question, please?
Of course. Can large-scale metabolomics projects be standalone without support from basic biological experiments and other omics approaches?
The thing about large-scale studies, metabolomics is very well suited for large-scale studies because, contrary to proteomics, for example, metabolomics can analyze hundreds and possibly thousands of samples. I would say, and I'm not an expert in proteomics, but I've done proteomics during my PhD, that it's relatively or quite difficult to do quantitative proteomics with hundreds of samples. So the ability to merge metabolomics studies with other omics, I would say that if the number of samples is very large, perhaps transcriptomics and genomics would be easier than proteomics. Proteomics, I think definitely there is an issue with the number of samples, unless we are talking about a targeted proteomic analysis.
Our next question for you. Is it difficult for systems biology in terms of numbers when one has to compare 10,000 transcripts versus 2,000 proteins versus 100 metabolites in terms of their collective overlap?
It is difficult, and actually I don't think there is a solution or a unique solution to integrate the information about thousands of transcripts, thousands of proteins, and hundreds of metabolites. We are trying to do it. As I said, we are collaborating with physics to do that. But I don't have a clear solution for that. I think everybody should find its own way. Our experience is that it is going to be, if one pretends to match gene expression with protein abundance and metabolite abundance, this will happen in very few cases. There will be a lot of uncorrelated data when trying to merge all this information, and the value is to understand what correlates and what doesn't and why. So it is a challenge, but soon we will see more studies in this direction.
Thank you. The next question for you. How does one obtain an accurate snapshot of metabolism without disrupting or stressing the biological system?
Please repeat.
Of course. How do you obtain an accurate snapshot of metabolism without disrupting or stressing the biological system?
Okay. Well, the snapshot of metabolism is what most of us are doing in metabolomics studies. So we are disrupting cells, tissues, we are extracting metabolites, and we are measuring hundreds of them at the same time. And we have information about, typically these are studies in which we compare a case versus a control, and we have information about the level, the relative abundance of these metabolites at one particular time point or phenotypic scenario. So that's for me a snapshot of metabolism versus the dynamic picture in which we can have a labeled precursor and follow how this metabolite is metabolized, is transformed into downstream metabolites at different time points and under different phenotypic conditions.
Great. The next question for you. How important do you see the linking of metabolomics data to biochemical networks and then subsequent informed targeting of genes as an important bridging of the omics gaps?
That's a good question as well. I think it's important. This is something I have discussed quite a lot with colleagues. The idea of understand biological systems from metabolites, from the bottom level of metabolites, from this cascade of this transfer of information that I was talking at the beginning of my presentation. So if we can understand metabolism, we can probably design better targeted approaches at higher levels. So we can probably target particular proteins and particular post-translational modifications associated with these proteins, and we can also study gene expression and the regulation associated with gene expression in a more targeted way. So this is something that, at least in our group, we are pursuing, and I think it's a valid approach.
Next question. Is there any hope for small isolated research group to advance the field or even survive in the field?
Yes, we do. We are a small group. If I can move my presentation. So our group right now is formed by 6 persons, including me. So I think we are surviving and we're doing well. So it's just all about good skills, motivation, and of course, one has to have good instruments, and that helps, and some money.
Thank you. Next question. What are your views on the use of model organisms such as C. elegans for metabolic flux analysis in vivo?
I do not have experience with metabolic flux analysis with organisms such as C. elegans. All our experience on metabolic flux analysis is on the field of cancer research, particularly using cancer cell lines. However, I know that it is possible to do flux analysis with microorganisms such as C. elegans. C. elegans, yeast. There are a lot of organisms that can be cultivated, that they can be incubated with a labeled precursor, that this precursor will be absorbed and metabolized by the organism, and eventually the trace of these carbons can be analyzed.
Thank you. Your next question. Do you think targeted metabolomics has higher capabilities to confidently identify more metabolites than untargeted approaches where many unknowns affect the data interpretation?
That's a good question. Targeted metabolomics has the ability, in my opinion, to quantify more accurately a particular metabolite as compared to untargeted. I mean, the analytical variability of triple quads, it's much higher than TOF or Orbitraps. Now, regarding identification, of course, when you do a targeted analysis, you have previously optimized the detection of a particular list or a particular metabolite because you have the standard. So you know the retention time, you know the fragments that you want to follow. So from that point of view, it's not about identification, it's you know what you're looking for. However, it is also true that there are certain isomers, and it comes to my mind, for example, glucose 6-phosphate and fructose 6-phosphate, which have almost identical fermentation pattern. And unless you optimize a very particular chromatography, they will also elute at very similar retention times. So in that case, I can see that either targeted and untargeted metabolomics may have problems, but targeted can probably design a very specific chromatography to separate these two isomers. But our experience with untargeted metabolomics is quite good, so we are able to identify relatively well a lot of metabolites as long as the intensity of these ions is good enough, and the fragmentation pattern can be matched with existing data in databases or by comparing with a standard compound purchased in any commercial vendor.
One more question for you. You mentioned the importance of mobile phases for LCM measurements. How can I know which salts additives eventually ion pairing reagent will help me to ionize my analyte, positive mode? I have not the time and the money to test them all.
No, me either. It's not about an empirical process of testing mobile phases or additives to these mobile phases. The example that I showed in my presentation came from an idea or something that I listened about using ammonium fluoride, about the basicity of fluoride in the gas phase. So fluoride, it has a high basicity. So it has a strong ability to catch protons. and leave molecules negatively charged. So with that idea in mind, I just tried to use ammonium fluoride for non-targeted or profiling experiments. Now, if there is any rational way to use additives for mobile phases, probably not, or at least I don't know. It's the list of additives or mobile phases that can be used for mass spec is also limited, because they have to be compatible with ionization.
Thank you. I think we have time for a few more questions, if this is okay for you, Dr. Yanis?
Yes, it's okay.
Perfect. Your next question. When you have to work with clinicians in order to look at the metabolomic profile, what would you recommend concerning the experimental design?
Well, the first thing that I would recommend is to pay a lot of attention to sample collection. So clinicians, some of our studies with clinicians haven't been successful. We just even started doing the study because the samples were collected at different hospitals using different protocols. And whereas this could be fine for genome sequencing, it's not the case for metabolomics. So if you obtain serum or plasma and you leave your sample in the table at room temperature for 4 hours before centrifuging, there will be a lot of changes occurring in this sample, and you may eventually compare samples that have been sitting in the table for 2 hours, others for 4, for 8, and you will see differences that are artifacts. So one of the main things that you have to pay attention is sample collection, sample storage, where all the samples stored in the same conditions, where all the samples collected in the same conditions. This is, for me, the first recommendation when doing studies with clinical samples. There are many more, but I don't think we have time to discuss all of them.
Of course. This will be the final question before we end our webinar. Do you think only using a simple MS instrument, example QMS, is sufficient for metabolite identification?
No. No, I don't think so. QMS, by QMS I understand single quads. Single quads are nominal mass instruments. So from that point of view, it's virtually impossible to identify a metabolite based on nominal mass information. It is true that you will also have, or it is possible to obtain the retention time on a chromatography for that particular compound. So it's still something, and it's still valid. So nominal mass and retention time, fine, but I would not consider that as an unequivocal identification.
Perfect. Thank you for your questions, everyone, and of course to our speaker, Dr. Yanis. Now, before we conclude our session today, I would like to advertise another great opportunity via the Metabolomics Society, and that is the 11th International Conference of the Society that will be held in San Francisco between the 29th of June and the 2nd of July this year. You can send in your abstracts by the 15th of March, and the theme areas, as you can see from your screen, are sure to appeal to a broad range of metabolomics researchers, and we are hoping many early career scientists. So on the next slide, I have been provided with a preliminary program from the local organizer of UC Davis, Dr. Oliver Feen, and as you can see, there are going to be several sessions across the 4 days of the conference. Dedicated to early career scientists will be 4 workshops, as you can see them highlighted in red. as well as a welcome reception the day before the conference officially starts. In terms of funding, the Metabolomics Society has two options available. The first includes 10 awards for registered students, and the second is another 10 awards for early career scientists that are within 5 years of their highest degree. Early bird registration ends on the 15th of April, so start getting those abstracts ready. As a member of the Early Career Members Network, I hope you will join us for our future webinars, and hopefully see you all in sunny San Francisco this summer. This concludes our webinar, and thank you very much everyone for joining us.